Now, let us look into other types of epistatic interactions. One is duplicative recessive epistasis. Duplicate recessive epistasis is the complete dominance at both gene pairs. However, when the either gene is homozygous recessive, it masks the expression of the other gene. For instance, in man, normal hearing is due to two dominant genes, capital A and capital B. But the recessive alleles small a and small b of the two dominant alleles are epistatic when they are homozygous. The paired recessive allele of one dominant gene masks the expression of other dominant gene. Thus, a person with the genotype small a, capital B would be a deaf mute because the genes small a are epistatic to the gene capital B for normal hearing. The paired recessive gene small b is epistatic to the gene capital A, which is responsible for normal hearing. This is a case of duplicate recessive epistasis, where either of the two recessive genes may be epistatic to the dominant alleles of the other. When a normal hearing person represented by capital A, capital B is crossed with a deaf mute represented with small a, small b, it produces a normal hearing person with the genotype capital A, capital B, small b in F1. If the F1 individuals are crossed themselves, they produce 9 normal persons and 7 deaf mute persons. Now, let us see about duplicate dominant epistasis. Complete dominance at both gene pairs, however, when the either gene is dominant, it masks the expression of the other gene. Independently, the ratio will be 15:1 if a dominant allele of both gene loci produces the same phenotype without cumulative effect. Complete dominance at both gene pairs, but the either gene when dominant epistatic to the other. For instance, in seed capsule of shepherd's purse, gene pair capital A, triangular shape is dominant over ovoid gene pair capital B, and the triangular shape dominant over ovoid produce F2 offsprings in the ratio 15 triangular 1 ovoid. Now, let us see about the biochemical basis of epistasis. Every cellular chemical reaction involves the stepwise conversion of one initial substance called precursor to another called end product. All the subsequent steps of a chemical reaction constitute the biosynthetic pathway, which includes various steps, and each step is catalyzed by a specific enzyme. Each specific enzyme is synthesized by a specific gene. Number of enzymes are involved in the conversion of one material to another material. In other words, series of genes are involved in the conversion of one material to another material. The simplest example to explain biochemical basis of epistasis is albinism. It develops due to the lack of melanin pigment, which produces black color. Melanin synthesis is not one step reaction like all other cellular chemical reactions. Rather, it is a 4-step process with each step mediated by a specific enzyme. These enzymes, which catalyze the reaction at every step, are produced by some specific dominant gene. We can get the idea of process with the help of the figure. The precursor is converted into the metabolite 2 only by the action of enzyme 1, and for the production of enzyme 1 the genotypic composition should be either capital A or capital A small a. The conversion of metabolite 2 to metabolite 3 occurs with the help of enzyme 2. The enzyme 2 is synthesized only when the genotype is capital B or capital B small b. The conversion of metabolite 3 to metabolite 4 happens with the help of enzyme 3, and the genetic composition required for the production of enzyme 3 is capital C or capital C small c. The metabolite 4 is finally culminates in the production of melanin, which happens with the help of enzyme 4, which is synthesized only when the genotype is capital D or capital D small d. As the gene capital C undergoes mutation, mutated gene small c could not produce enzyme 3. No production of further metabolites happens, and the result is albino. This is illustrated in the figure given in the screen. Now let us examine about duplicate genes with a cumulative effect. This could be additive genes or polymeric genes. Two independent dominant genes, whether present in heterozygous or homozygous condition, express similar phenotypic effect when present individually, but produce a double effect or a cumulative effect when found together. In other words, two completely dominant genes controlling a character produce identical phenotypes when they are alone, but both genes are present together. Their phenotypic expression is enhanced as the effect of the two genes were cumulative additive. In additive gene action, each dominant allele of the two genes produces equal and identical effect on the character. The effects of the all dominant allele at the two loci are additive, that is, if the dominant condition at either locus produces the same phenotype, a 9:6:1 modified dihybrid ratio is obtained in the F2 generation as phenotypes produced by single gene dominance of the two different genes are similar. In this given table, you can see the mode of action of duplicate genes with cumulative effect. The first column represents epistatic alleles, second hypostatic alleles, and third phenotypic expression of alleles. In the first row, the epistatic alleles is capital A small a, hypostatic alleles capital B small b. The phenotypic expression is neither small a or small b. In the second row, the epistatic alleles is capital A small a, the hypostatic alleles is capital B or capital B small b. The phenotypic expression of alleles is capital B only. In the third row, the epistatic alleles is capital A small a. The hypostatic alleles is capital B small b. The phenotypic expression is capital A only. In the fourth row, the epistatic alleles is capital A small a. The hypostatic alleles is capital B small b. The phenotypic expression is capital A and capital B. Another example would be fruit shape in summer squash, Cucurbita pepo. Certain species of summer squash can exist in different shapes. They are long, spherical and disc shaped. In case of Cucurbita pepo, the fruit shape is controlled by two genes, gene capital A and gene capital B. The disc shaped summer squash has both the dominant allele capital A and capital B, with a genotype capital A either with capital A or small a, capital B either with capital B or small b. Elongated summer squash produced when both genes are in recessive combination small a, small b, small b. Spherical summer squash has either of the two dominant genes capital A or capital B with a genotype capital A could be with capital A or small a, capital B, or small a, capital B with capital B or small b. When two different races of sphere shaped summer squashes with genotypes capital A, small b and small a, capital B are crossed, the F1 progenies are all disc shaped with the genetic constitution capital A, capital B, small b. When these F1 progenies are self pollinated, interaction between both the dominant genes give new phenotypes in the ratio 9:6:1, wherein 9 disc shaped are formed, 6 sphere shaped are formed and 1 elongated shaped. Epistasis or interactions between genes has long been recognized as fundamentally important to understanding the structure and function of genetic pathways and the evolutionary dynamics of complex genetic systems. With the advent of high throughput functional genomics and the emergence of systems approaches to biology as well as a newfound ability to pursue the genetic basis of evolution down to specific molecular changes, there is a renewed appreciation both for the importance of studying gene interactions. Epistasis was originally a concept from genetics, but is now used in biochemistry, population genetics, evolutionary biology and computational biology.